So hopefully you can hear me. Again, welcome to the CADASIL Connection webinar series. And on behalf of myself, I'm Bert Kosiski, president of cureCADASIL. And I'm here today with Vinita Ball, who's one of our board of trustees and actually sets these webinars up for us. And we're pleased today to have our guest speaker, Elisa Ferrante, who's going to be talking about the CADASIL clinical studies at the National Institutes of Health. In the room with Elisa is Dr. Manfred Burham, who is also an NHLBI. Oops. So cureCADASIL is a nonprofit organization. Having a little trouble with slides. Is a nonprofit organization dedicated to helping patients, families, caregivers, and other supporters touched by CADASIL. We aim to unite patients and the medical community toward the common goal of treatments and ultimately a cure for this rare genetic disease by promoting communication, advocacy, research, and education. So our presenter today is Dr. Elisa Ferrante, and she is the clinical program manager and staff scientist at the National Heart, Lung, Blood Institute at the NIH in the Translational Vascular Medicine Branch. And her background education is actually her BS degree was at University of Virginia in mechanical engineering, her master's in biomedical engineering, and then ultimately her Ph.D. in biomedical engineering. Dr. Ferrante will discuss the significance of clinical studies of CADASIL that are being conducted at the NIH. She will also provide updates on the NIH CADASIL pilot and CADASIL Disease Discovery Studies, which are aimed at understanding the development and progression of the disease. In their studies, they are continuing with the 20 previous participants in a pilot study that is now completed and recruiting additional CADASIL participants. I believe their goal is 100 total to learn more about how CADASIL affects a person's blood vessels over time. Control subjects are also being included. So a technical note here, please note that audience will be muted during this presentation, but we'll take questions at the end. If you have questions, you can type them into the question box in the drop-down menu, which should be on the right-hand side of your screen. And then at the end, when Dr. Ferrante is done with her presentation, we will have open, and I think there'll be plenty of time for questions, and we'll take them one by one. So without further ado, I am going to turn it over to Dr. Ferrante, and we'll so
can you guys see my presentation?
yes.
Yes. Excellent. Let me just make some windows a little smaller so I can also see my presentation. Okay, so I am Elisa Ferrante. I am a staff scientist here at National Institutes of Health. And I work with Dr. Baum, who's in the room with me, but will join later to help us answer questions, as well as Dr. Toro, who is the neurologist in our CADASIL team, and will join us around 11:30 to be able to answer questions as well. You can see here the NIH Clinical Center in the picture. This is part of our large campus in Bethesda, Maryland. And this is where patients come when they enroll in our protocol. This campus holds about buildings for about 27 different institutes. Some of them study different things. So for example, NCI is the National Cancer Institute, NEI is the National Eye Institute, and in particular, we are the National Heart, Lung, and Blood Institute. You'll hear me mention NHLBI is our acronym. And so we really are looking at CADASIL with the lens of what is happening to the blood vessels, not only in the brain, but throughout the body for people that have variants in the NOSH3 mutation. This building that you see in the front is actually the clinical center. And here's another picture of it. The clinical center is where patients come to participate in our studies, and every patient that comes is enrolled in either one or more of our research protocols. So this is truly just a research hospital, and it is actually the largest research hospital in the United States as well as the world. We do have cutting edge technologies, imaging, instrumentation. We provide great care to our patients, but we don't have the ability to bring patients just for urgent care or, for example, labor and delivery. We don't have those departments here. You'll see just in 2023, we had just over 6,000 patients visit the NIH participating in one of our many clinical studies. We have about 1,500 active clinical studies, and many of our patients do come in for inpatient visits. We have about 200 inpatient beds that can accommodate that, but the majority of our patients are here as outpatients. And in particular, our CADASIL study is for people coming in outpatient. They would be visiting one or a few of our many outpatient clinics, and they would be meeting with a pretty diverse team of clinicians, Ph.D. researchers like myself, and other clinical staff like nurses, phlebotomists, and different techs with different specialties. And as I mentioned, all of the patients have to be enrolled in a clinical study, which means all of the patients are volunteers participating in our research. So really, patients are part of our team. And the care that they receive at the clinical center is protocol-centric care, which means that it's very tailored to that specific patient and what they have going on when they come and see us. All of the care is received at no cost to the patient, and we enroll patients from across the United States as well as internationally. Travel is paid for inside of the United States. And the goal of the clinical center is really to use science to advance or improve human health. So in that context, we're a leader in bench to bedside medicine or translational medicine. And actually one of the favorite things my favorite things here at NIH is the way that the building is set up. So the clinical center has all of the patient clinics and patient facilities in the very center of the building. And the research labs are on the outside surrounding the patient areas. And the idea is that we get the information from the patient to help us inform the research we do in our labs, but then the lab also makes its discoveries and then informs what is happening with the patient and the patient care down the line in this true bench to bedside concept. So as I've mentioned, the mission statement of the clinical center is to provide hope by pioneering clinical research to really improve human health. We focus on several guiding principles that revolve around compassion for our patients and their families, with diversity and inclusion for people and ideas at the forefront. We're also looking to provide high reliability in delivering patient-centric care to our patients with excellence, accountability, commitment to our people that are working here for professional growth and development, and ultimately to push innovation to prevent and solve problems in the healthcare setting. But then the NHLBI mission further goes into providing global leadership for research training and excellence education programs to help promote, prevent, and treat heart, lung, and blood diseases in a way that enhances human health of everyone so that they can live longer but also more fulfilling lives. And I don't think this is in the radar for a lot of people. I learned of this documentary. It's a 3-part documentary made on NIH back in 2017. It was made by Discovery, and I got to see it premiere here at NIH. So I didn't have to have a subscription to a particular channel, but if you do, you may have access to this very cool documentary that is really a good example of the mission work that we do here at NIH. And I do have, let's see, I should be able to show you the video here. It looks like I can't, guys. I know we tested this, but I don't see the video. Oh, there it is. Okay, so this might show up as a smaller window for you. You can make it larger by expanding the window on your computer. And let me go back to my presentation, which hopefully you guys can see again. Okay, so with all of this background and frame that exists to support this clinical research, we started the first natural history CADASIL Disease Discovery Protocol. And for this research, we assembled a fairly large team of investigators throughout the NIH, so across many of our different institutes, because we were interested in looking at different components of CADASIL. And we also have some extramural collaborators that are helping us figure out what is going on at the molecular, at the cellular level with CADASIL. And so you see here at the top is our core team here at NHLBI. We also have a lot of support from clinical staff as well as research lab. And on the right side, you see we're very focused on the neurological component of CADASIL. So we're looking at imaging, we're doing neurological consults and psychometric testing, but we're also focused, as you see here on the left side, on vascular testing and understanding better what is happening with the blood vessels in CADASIL. Okay, so if you're here, you probably know a lot about CADASIL already, so I'll be very brief on this. But CADASIL is caused by genetic variants on NOTCH3 gene. They can span any area of the gene, and so they will translate to any area of the molecule NOTCH3 that is expressed on smooth muscle cells. And here in this schematic, it's abbreviated SMCs. And This can only be diagnosed really well and consistently by doing a genetic test. Previously, the gold standard used to be taking a skin sample and looking for these little deposits. They're called granular osmophilic material, or GOMS, outside of blood vessels. And this is a blood vessel image that I took with an electron microscope of one of our patients that has CADASIL. And it was we used a skin sample. But we don't know exactly what is in the GOMS materials, and we don't know exactly their role. We do know that the dysfunction in the NOSH3 molecule causes those deposits, and also eventually endothelial cells, labeled ECs here, to start breaking down the barrier inside of the blood vessel, and you end up with leaky blood vessels. So it is a small vessel disease. It particularly affects the brain, although it can also have effects in other blood vessels throughout the body, which is why we can see the GOMS in skin samples. And as far as clinical symptoms, it is a slow onset disease. It's an adult onset disease, and people in their 30s and 40s start developing recurring migraines that can have an aura or not. And this may eventually lead to strokes or transient ischemic attacks, TIAs, and it could down the line lead to cognitive changes. Now, because we don't know the molecular mechanism exactly, we also don't have available treatment options that are specific for CADASIL, although we are able to treat the symptoms. And so that brings us to our first CADASIL Disease Discovery Protocol. This started in 2016, which is also when I came to NIH. So it's been very exciting being part of this from the very start. And it ended in 2022 because of the COVID-19 pandemic. It was a little bit later than we expected, but the clinical center closed to keep our patients and our staff safe. This is a natural history protocol, so we're just studying the disease. And the goal was to enroll 20 patients that came every year for about 3 years. Our primary objective at the time was to better understand the molecular disease mechanism, so what's happening at the cell level. And we were also looking to see how that caused the clinical symptoms that we were seeing in CADASIL, with a secondary objective of trying to figure out why what the different clinical symptom variations were. So we knew that there were different symptoms that different people were experiencing. For inclusion criteria, we only enrolled adults. They could be male or female, but they had to have an established diagnosis of CADASIL from genetic testing. And what was key for this pilot study is we were looking to have the first study visit within 5 years of people being diagnosed, which translated to people having mild or no cognitive impairments. We were not able to enroll people that could not give informed consent, either themselves or through a legally appointed representative. We could not enroll pregnant women, though pregnant women or women can participate in the study after they give birth. So we just ask people to wait. And what is really critical is we cannot enroll people that can't get an MRI scan. And that may be either because of an implant that they might have that can't go in the scanner, or because of severe claustrophobia. But the reason this is that the brain MRI is really a key feature of CADASIL, and is something that we measure as one of our primary measures. So you can see here the breakdown of each of the visits when a person was coming in to the clinical center. Visit 1 was pretty heavy. There's a lot of checks there, but it's really because that was our get to know you visits. And then there's a little bit less at visits 2 and 3. We focused on really understanding the medical history, the family history of our patients. And then we did a physical exam with vital signs and clinical blood work on EKG. For the neurological components, we were looking at the brain MRI. We also had an MRA at the time to look at blood vessels, and a neurology consult with Dr. Toro, who will join later. And we do a very extensive neuropsychometric testing set. It's about 13, 14 tests that are trying to break down the different cognitive domains and see what's happening there. For the vascular testing, there are a lot of individual tests listed here, and I will not explain all of them, but just on a general level, we were interested in looking at blood vessels throughout the body. And so those measurements were an OCT, cardiovascular ankle brain index, pulse wave velocity. But we were also very interested in looking in the eye and looking at the retinal blood vessels, which is why we have a fluorescent angiogram included here. And lastly, for the lab research side, we were collecting a small skin sample as well as a research blood that would allow us to make cell lines or study what is happening in the blood and skin of patients with CADASIL. To give you a little bit of a window on our patients, we actually ended up with 19 patients, and that is because one of our patients, we identified an additional diagnosis that had symptoms overlapping with CADASIL, and so it was a little too difficult to parse out what was CADASIL and what was the other diagnosis, so we had to exclude that patient, which meant we lost one of our male patients. But generally, we try to have an equal number of males and females because we don't know yet if there is a sex component to help people develop CADASIL. Our population is largely white, although we know that CADASIL can happen in any race and ethnicity. And then we look at medications and enrollment as a way to figure out what people are getting treated with and what they have going on at the baseline. So we're interested in mood disorders, neurological components, but we're also looking at the cardiovascular risk factors. And so we're interested in understanding what people are being treated for there in the context of cholesterol, hypertension, blood pressure, diabetes, smoking, and so on. When we looked at our genetic background for our patients, we see a fairly wide distribution. So you can see here that exon is a portion of the gene, and our variants happen in many of the different exons of the gene in NOTCH3. And there is a little bit of a cluster here on exon 4, and I want to point out that it was a very small patient number, but we only had actually one set of siblings. So we aimed at having people that are not related to each other so that we wouldn't have the same variant over and over that we were studying. We didn't want to bias our genetics. And so diving into the results in a little more detail, one of the things, the first thing really that we did was look at the brain MRI. And you can see here, we have a typical brain MRI for CADASIL patients, where you see the white matter changes. That's these white portions in the gray brain area. And what we're doing is we're using artificial intelligence to develop machine learning methods that allow us to create these masks in red for the white matter changes, but they help us track over time what is happening there. So you can see here between visits 1 and 3 that we're able to use these methods to detect, for example, in this area, even very small and subtle changes over time in the brain of patients with CADASIL. And just a cohort at a glance as far as neurological components. A lot of our patients came in already with strokes and transient ischemic attacks. Some of them did not have it and developed it later, and some of them didn't develop any strokes at all during the study period. We also looked at cognitive impairment, and there was some mild cognitive impairment in our patients. A lot of them did have migraines as a symptom. And then we looked at mood disorders, so anxiety, depression, things like that. And some of that was also happening, even at the early stage of the disease within those 5 years of diagnosis. And the neuropsychometric testing, I will say there is a lot of data there, so I'm only showing you guys a glimpse. But you can see here the different domains, the different cognitive domains that we're looking at, as well as an overall average, where this particular group of patients is doing very well, with already a pattern that we can observe in the psychomotor domains, so like fine motor movements, for example. So as a summary to this pilot study, we saw pretty slow progression over the study period, which was short. It was only 3 years. We see the typical white matter changes of CADASIL in all of the patients that enrolled, regardless of where their Notch3 variant was. And very exciting, we're able to measure that. Our genetic background was very heterogeneous, and we saw that there was also a variable clinical symptom presentation, with some patients having no symptoms at all and some patients having more significant symptoms. For the cognitive side and neurological side, we saw that balance, verbal, visual learning, and the psychomotor domains were the most affected, even at the early stage of CADASIL. And there is a lot more data, but it is currently in analysis. And we hope to be able to share that with you soon as we get publications out, hopefully within the next year. So we'll make sure to send those to cureCADASIL so they can let you guys know as well. We learned a lot from the pilot study, and we wanted to learn more. So we decided to continue studying CADASIL in a larger CADASIL study. So we expanded this to be able to enroll about 100 people that have a Notch3 variant. It is still a natural history protocol, and we did invite the original 19 people we had to enroll, and we got some of them to come back because I think they like us. We also were able to add healthy volunteers. So we are enrolling up to 40 healthy volunteers that are more or less age and also sex matched to our CADASIL patients. And we really extended the study period to go from 3 years to 9 years because of the slow progression. And so these patients are now coming to the clinical center for 4 visits that are about 3 years apart. But we ask that they let us know if something major happens between visits in those 3 years so that we can continue building our knowledge base on CADASIL. We fine-tuned the primary objective, so we're still looking to understand the molecular mechanism, but now we're also looking at the progression of CADASIL so that we can understand better how fast or how slow or how different people develop CADASIL. And for our secondary objective, what we're really interested in doing is looking at the genetics and the clinical symptoms and really trying to find links on what is happening there. So maybe someone with a Notch3 variant in a particular location has different symptoms than those with a different Notch3 variant. We're really trying to make those connections. Our inclusion criteria is largely the same. We still do adult males and females, but we've also added healthy volunteers, which is exciting. And we still need a genetic test of CADASIL, but we dropped the 0 to 5 years from diagnosis to be able to be more inclusive. Our exclusion criteria remains the same. We also fine-tuned our study visits. So our study visits now take between 3 and 4 days. It was more or less the same at the pilot study, but it was a really packed schedule. We are still looking at medical history and family history and doing a fairly comprehensive physical exam with clinical labs and EKG. For the neurological component, we do still the neurology consult and neuropsychometric testing, as well as the brain MRI, but we've added a 7 Tesla brain MRI, which we do later in time. And that is for us to be able to look at the brain in more detail. So the 7 Tesla gives us better resolution. For the vascular component, that is also a trimmed down set of testing. We are still doing the eye exam because we're very interested still in the retinal blood vessels. And we've added a couple of non-invasive tests like the near-infrared spectroscopy, or NIRS, that is looking at how our blood vessels in the hands and fingers react to an occlusion with a pressure cuff. So we can see how well they expand and contract in response to lack of oxygen. And neopholcapilloroscopy is an interesting test. We're actually putting a miniature microscope at the nail fold, and we can see individual blood cells moving through the capillaries in this test. And we can use that to compare to healthy volunteer capillaries as well. For the biospecimens, we have now an optional lumbar puncture. So we're interested also in identifying biomarkers for CADASIL, and I'll speak on that a little more later. But our patients, if they're willing, can also give us spinal fluid, so we can do some of that work. To date, we have enrolled about 21 patients with CADASIL and 9 healthy volunteers. Something else that is very exciting that has happened in the last couple of years since we started with the pilot study is other groups, not only in the United States, but internationally, are starting to look at CADASIL in more detail. And we've been actively establishing collaborators with different groups so that we can have a larger group of patients with also more diversity. And that allows us to get to solutions faster when we're collaborating with other people. And the work is ongoing. And I also wanted to highlight what is happening on the molecular side. So our extramural collaborator, who's at Northwestern University, that worked with us since 2016, recently published a study on Notch3. Her name is Dr. Rivera Rispe. And what her team found was that in a mouse model, Notch3 already has some loss that happens on the blood vessels, and that this does lead to changes in vascular contractility that can lead to neurodegeneration over time, which may be something that is translatable to human patients. And I have also here a video that I can show that kind of summarizes her findings. Let's see. Okay. And so we've made some strides on the molecular side, as well as, bear with me for a second. we've learned a lot from our CADASIL natural history studies. So to summarize, these natural history studies are really critical for us to build our knowledge base on how the Notch3 variants may or may not lead to CADASIL symptoms. And we still need to understand the disease mechanism, but we're also very focused on finding biomarkers, which are circulating molecules in your blood. They can be found in spinal fluid, for example, because not only might they allow us to track disease progression over time, they would also be very important as endpoint readouts if we do find a treatment. And without these natural history studies and building this knowledge base, it is very difficult for us to get to the treatment, which is why they are critical. At the end of the day, a lot of our patients also go through a journey in getting diagnosed with CADASIL. So there really needs to be more awareness of CADASIL, what it is, and how to diagnose it, so that we can help our patients earlier when they start developing symptoms. And what is key is our study participants who are part of our team and central to everything we do, as well as these collaborative research partnerships that are starting to pop up and are very exciting. And to close, I wanted to share our study information. This is actually one of the flyers that we share with people. I know there are links on my slide that you cannot click, but if you scan the QR code on this slide and the next slide, it'll take you to the study website. And this slide also has my email address. You can send me questions and reach out, as well as our patient recruitment center here at the clinical center. So anyone who has CADASIL or Notch3 variants and is interested in participating can call there, and everyone gets referred back to us. And to close, I want to thank cureCADASIL for giving us this platform to share with you what we do here at NIH. But also thank you for participating and all of our patients for being part of our clinical studies.
So Elisa, thank you very much. This is a great presentation. I'm particularly glad that we have time for questions because there are a number of questions that have been coming in. And maybe I'll start with a few of the more practical items that people are asking about getting into your study. And the first one was, what does the process of enrolling and coming to the NIH really look like?
Yeah, so anyone who's interested in participating in our study would either reach out to the recruitment center or the contact information on our website. And once they reach out, someone from our team will connect with them. They'll go through the eligibility criteria and see if the person can participate in our study. And once all of that is green lighted, we bring the patients in. So we'll have a patient care coordinator who reaches out to the patient to determine dates. And then once we have those, we build the visit testing for that patient. We send out a schedule, and then eventually we bring them in. They come in for a visit that is between 3 to 4 days, depending on how long they want to be here. We're very close to DC and all of the Washington, DC sites as well. And the patient would be outpatient, so they would be able to stay either in the Safra Lodge, which is like a hotel that we have here on campus, or they can stay at a hotel nearby. And then something that I wanted to mention earlier, but I want to mention now, is all of the clinical data that we collect here at NIH is also shared with the physician that follows that person on the outside, so that they can help guide that clinical care as well. On the research side, that data takes a little longer to crunch and analyze, and so that comes out in the form of papers later in time.
Great. Here's another question. who can be a healthy volunteer? Could my partner sign
Um,
up if they accompany me to the NIH?
Yeah, so oftentimes our patients are accompanied by their partners, and so we have wives and husbands that participate. We can also have relatives that have a confirmed genetic test that tells us they don't have the Notch3 variants. But really anyone can participate. We're just looking for adult patients that kind of fit the CADASIL age group.
So a related question, can you do the genetic testing for diagnosis? So if somebody comes as a partner, for example, and they haven't been tested?
We are not able to do the genetic test here, and that is because that genetic test needs to be ordered by the neurologist or the PCP on the outside. And there is an amount of genetic counseling that happens through that process as well. And so we don't have the ability to do the genetic test here, which is why it is critical actually that anyone who's interested in participating be able to share the genetic test with us.
Here's one: I'd like to help but don't want to come to the NIH. Is there something else I can do? Like send blood
Yeah,
samples?
yeah. Patients with CADASIL that might be interested in helping the research but don't want to come to NIH could potentially send us blood samples. They would still need to be enrolled in a different protocol that we have where we build a repository of biospecimens. But that would be, we have a kit that we can send out and someone can go to a Quest clinic anywhere and get the blood draw and the samples come back to us.
There are a couple of questions here. I tried to put these together about other studies. Okay. So one of them was, can someone participate if they are also enrolled in other CADASIL studies? And can you use the data collected at different places? And then a related to that question, and I'll give this a related question because they're very similar.
Yeah.
A related question is, why are there multiple natural history studies like the one that you're doing?
Okay, I'll start with that question because it's, kind of the basis. There are a lot of different groups that are studying CADASIL, but they're looking at it with different lenses. So some people may be more interested in just the brain MRI. Some people may be more interested in a different organ system. And so everyone is kind of focused on a different thing, which is why there are so many popping up. So everyone is doing a little bit different. We're focused on the blood vessels, for example. Yeah, people can participate in multiple studies. We don't have a restriction on that. The only thing about sharing the data is the brain MRI, for example, is a critical measure for us. We have a very tailored sequence for the brain MRI that helps us look at the blood vessels. And so there are differences between sequences as well as differences between machines and who makes them that make it hard for us to share that type of data. And so when our patients come here, they're measured on the exact same MRI scanner even, so we don't switch them, and we use that exact sequence at every visit so that we can keep track of what is happening there. But there is other data that can be shared, particularly biomarker stuff and some of the other tests.
I think you started to hit on this a little bit. Here's a question. First off, thank you for this wonderful webinar. Will the NIH study participants have access to the biometric/clinical data collected?
Yeah, so all of the data that is clinical, that can guide patient care is yours. And so that data, the patient can choose to share with their doctors on the outside to help guide care.
You mentioned cell lines were being collected, and there's a question here: how many cell lines, and if you're referring to iPSCs here, and how many of these cell lines now have NIH collected? I remember that I think you had a publication a while back, and there were 5 cell lines at that time, but
Yeah, we collect blood and we collect skin, and from those samples we generate cell lines. We start with fibroblasts, and from that we can make iPSCs and turn them into either smooth muscle cells or endothelial cells or other types of cells. So we published on 5. I believe we created 10. And we're still continuing to do that research so that we can build an in vitro patient model in the lab to be able to really study that molecular mechanism. And once we have that molecular target, to be able to test different types of treatments of maybe drugs that are already available and can be used off label, for example. And we'll also eventually use those for in vivo studies and animal models. But we're also doing a lot of work on something called an organoid. And so we're developing vascular organoids in the lab that will allow us to really study the blood vessels and that interaction between the smooth muscle cells and endothelial cells. And hopefully we'll have some publications coming up on that in the next couple of years.
here's another very specific question. Have you found anything interesting with the retinal imaging component you mentioned?
We're doing a lot of testing that is similar to the testing that other people are doing in the other studies. And we're looking at what the peripheral blood vessels look like and what's going on there. We think the data is interesting, and we hope to have that publication out very soon as well so that we can share with people.
Another question related to the multiple studies that are out there. And this question is, with all of the CADASIL studies going on, is there a meta-analysis that can help us collate strategy data, especially as it relates to race, ethnicity, sex, and gender? Meta-analysis.
Yeah, so I think a lot about like the distribution as far as race, ethnicity, for example, and I think that a meta-analysis would be very, very exciting. And for that, we're setting up these collaborations with the different groups within the US, but also globally. So there are groups in Asia, there are groups in Europe that we're talking to. And we don't have a specific strategy quite yet because those conversations started sometime last year, but the hope is that we can make that happen.
Cool. Another very specific, have you found plasma notch 3 extracellular domain as a specific biomarker?
I'm still looking at the biomarker data. I have not seen it pop up in the analyses we have done so far. I do know that the GOMs, those granular osmophilic materials that you see outside of blood vessels and the skin, have cleaved Notch 3 extracellular Notch 3. So it does deposit there, which is maybe one of the reasons why we're not seeing circulating. But yeah, I have to do more in-depth analysis on the biomarkers to better understand that.
So I'm going to put you on the spot here a little bit. You know, biomarkers and potential biomarkers that down the road might be surrogate endpoints or outcomes for clinical trials. What, based on all of your experience, and I think Dr. Burnley is in the room too, what is your best guess here? I mean, what is where is the money at? I know there's a lot of controversy, for example, over MRI findings and changes. Some think that that will be the way to go, and others think that that's not going to happen. What are we going down the right path here? What do you think we'll find?
I'm not really sure. I think the brain MRI is very useful, particularly as far as diagnosing CADASIL because it has a specific way it looks. And people need to learn more about that for sure. Without really having gone in depth with the data analysis for the biomarkers, for example, it's really hard to know what's going to happen. But that's why these natural history protocols and studies are really key because the problem is we don't know enough and we need to learn more.
Another question here. Are you looking at disease progression with the particular medication study participants are taking?
We're collecting about 100 people, so we'll be able to see what the different medications are that people are on. And hopefully we can look at different groups within that patient cohort. So that is something that we'll keep in mind.
I almost hesitate to ask this one because I don't know, but there is a question here. Can you show slide 22 again? And so somebody must have had a question about slide 22, and I do not recall which slide that is.
Slide 22 is a backup slide. I didn't really show it.
Yeah, I don't know who's asking
Yeah.
that.
I mean, this is some, yeah. I had this in the original presentation and I decided not to put it in there. But this here is your NOSH3 molecule on a smooth muscle cell, and this is the JAK1 receptor that it binds to.
Anyway, so I think this will be a good spot to end. I don't see any other burning questions out there. I'm going to bring up my last slide here in a second. Been having delay problems here on the internet. So whenever I do something, I have to wait a little while. Well, my last slide was basically a thank you. This is an excellent presentation. And also to acknowledge that the recording will become available. Usually it takes a day or two before we get the recording up, but there were a couple of questions about that. And so, yes, the recording will become available if people want to go back. And also, if there are people who are unable to actually make it to the webinar. So on that note, I think we'll call it a day. And again, thank you, Elisa, and all the effort you want to put into this. You could tell by the questions that there's a lot of interest in your study, and I'm sure that people will be knocking down the door to enroll. So
thank you, guys.
Thank you very much.